Hello everyone. What we're going to be discussing in this screencast today is what you're going to be doing in this first week of these 4-week experiments, which will take place from week 8 to 11 of your Bio 2 course, where you learn basically how to isolate bacterial genomic DNA. So first, you'll learn how to prepare the bacterial media and grow the bacteria, that's all we'll do this week. And then you'll see in the following 2 weeks, you'll learn how to basically collect the bacteria that you have grown in the week before and how to extract the genomic DNA from it. Finally, on the 4th week, we'll learn how to also quantify the genomic DNA of these bacteria. So, what you're doing this week is you learn how to grow the bacteria. In order to do that, you basically have to do 3 things. First, you will have to prepare and sterilize a bacterial growth media. This is basically a solution or a media itself, which basically provides everything you need to grow bacteria, to grow large numbers of bacteria. Once you've done that, then starting from this media, you'll prepare what we call the agar plates. So basically, you will pour this media into some containers, which are the plates or the petri dishes, where you will let this media solidify. And finally, once the media will have solidified, you actually add the bacteria to this media to let it grow overnight. So you're going to do 3 things. You'll learn how to prepare and sterilize the medium, how to basically cast it inside these petri dishes, and finally, how to add the bacteria to this media to get it to grow overnight. So, what is bacteria growth medium? As we said before, it's basically a solution or a media that provides all the nutrients for bacteria to grow very well. Now, if you had it to prepare yourself, what you could do is simply add some trypsin to some extract from yeast, and that's an easy way to produce it. By trypsin, what it is basically a digest, so it's a series of protein caseins from milk, which is digested with an enzyme called trypsin. And so basically this digestion generates a series of short peptides and amino acids, which are very helpful for obviously the bacterial growth and formation of protein, stuff like that. To this, you can add basically an extract from yeast, which you will do by growing yeast and killing them basically and drying them. And this mixture here from the yeast will contain all the other necessary molecules like amino acid, vitamins, nucleotides, and so on. So by mixing these 2 components together, trypsin and an extract from yeast, you will actually have a media which will sustain very, very well the growth of bacteria. Now, this can be obviously very lengthy. If you have to take these proteins, digest them, take yeast, grow it, and so now, fortunately, all of these media can be simply purchased from different companies, so they're available commercially. They've already been made by different companies, and one example is the one you'll use today, which is called the plate count broth media right here. Now, when you talk about bacterial growth medium, in general, there are 2 different types that you can use. You can use a liquid medium or a solid medium. Basically, the medium is exactly the same. The only difference when you talk about a solid medium is just basically a liquid medium to which you have add a solidifying agent, which is agar, a product from seaweed. Now, the difference though is in the way the bacteria will grow. So if you use a liquid medium, you usually can get a much larger number of bacteria. Why? Because if you add bacteria to a liquid medium and you shake this medium, these bacteria can actually diffuse throughout the liquid. So they can actually grow on the surface or in all directions inside this medium, and basically they will grow in a 3-dimensional way. So the entire medium will be filled with bacteria after a certain period of time. Now, if you use a solid medium instead, and you add the bacteria on top of that, this media will be casted first of all, before we say how they will grow, they will be casted inside what we call the petri dishes, which consists of basically 2 pieces of plastic, one larger than the other. The one that is larger is what we call the lid, and the other one is the one where basically the growth media will be solidified inside, right here. So now if you add, after this media has been solidified, which is what you're going to be doing this week, if you add the bacteria on top of that, the bacteria now at this point can only grow on the surface of this media. So they can only grow basically in a two-dimensional way. And obviously this will produce much less bacteria compared to a liquid medium, but it has its own advantage, which we will not discuss in this screencast right now. This is just a series of pictures that shows you the two different type of media. So here on the left, on the top left, you see an example of a liquid medium which does not have any bacteria growing inside, so it's nice and clear. And here you see a plate with a solid medium, bacterial medium. Now once you add the bacteria to it, you can see that the liquid medium here will have bacteria growing throughout and making the entire solution here completely opaque. Okay, so bacteria have grown in a three-dimensional way in this case. Whereas if you add bacteria to this plate, in this case, for example, just a few bacteria being added to this plate, you see that the bacteria basically will grow into small colonies. If you add enough, actually will cover the whole surface. But basically this colony here, they all consist of one original bacteria which has multiplied into millions of bacteria which stay nearby it, and so will form this visible white column made of thousands of bacteria, but again all originating from a single bacteria growing there. So in this case the growth has been two-dimensional, only on the surface of the plate. Now, how do you prepare your media in this experiment? So first of all, you will use the plate count broth, so this commercially available mixture of nutrients for bacteria. And what you have to do basically is prepare a solution for 5 different petri dishes, with each petri dish needing basically 25 milliliters of medium. So since you have 5 of them, you will need to prepare basically a 125 milliliter solution, which will contain the plate count broth in a concentration of 1.7% weight over volume. This is the medium itself. This is the nutrients the bacteria need to grow. And to this solution, you'll add also 1.5% of the solidifying agent, which is agar. Now, once you do that, you'll see that the plate count broth will dissolve fairly easily, but you will notice that agar will not dissolve. This solution will not be homogeneous. This solution, by the way, has to be prepared in a 500 milliliter Erlenmeyer flask. So now you have mixed your medium, bacterial medium there, and the solidifying agent, and now you have to get it ready to be poured inside these plates to form the solid media right there. Before you do that, however, you want to make sure that all the bacteria that come with the flask, for example, with the media itself, these are non-sterile components. The agar itself is non-sterile. So each one of them carries some bacteria which are not the bacteria that you want to grow. What you're going to be growing is E. coli, Escherichia coli. You don't want to have any other contaminants coming from anything else. So basically what you have to do is you have to sterilize this solution that you just made here before you actually put it on the plate. And so the way you'll do it by doing what we call the autoclaving. This is a process where you sterilize any material by treating it with steam at high pressure. So steam at 121 degrees Celsius at a pressure of 15 psi. And this treatment can be done for 15 to 20 minutes. We will use 15 minutes in our experiment. So by putting the media in this autoclave at steam at high temperature and pressure, you'll actually basically kill any bacteria which is existent there. Another advantage of using the autoclave is that the high heat will actually help the agar dissolve as well. So you will notice that the solution that you made will go from a non-homogeneous solution to an homogeneous solution from before and after the treatment with the autoclave as well. Now, after autoclaving, you cannot pour the liquid on the plastic yet. It actually will melt or it will affect the plastic itself. So first you have to let it cool down to about 55 degrees. How do you know you reached 55 degrees? Usually that's a temperature where you can actually hold things on your hand. Well, it will not be too painful anymore to hold in your hand. So from time to time, what you have to do is take your flask, put it on the palm of your hand, and feel whether you can hold it or not. If you can hold it at this point, you can assume it's about 55 degrees. Once you have that, actually before you do that, while you let it cool down, now you have to take a plate. And so if this is your plate here, this will be, and let me draw it here. I forgot to add a picture here. You'll have a larger part outside, one inside. So now on the smaller part, on the bottom of it, you have to write down a series of information which will allow you to then figure out which plate is yours later on. So you have to add the date, right on the side, the date, your name here, the organism you're growing, which is Escherichia coli, and your lab section here, which will be bio 273 CG, depending on your section that is. Okay, so you write around the plate while the media is cooling down. And once the media is cooled down, now you're ready to put it inside there so that you can actually form this solid media inside there. Before we talk about how you do it, what you have to do is turn on what is called a Bunsen burner. This is an instrument which is very commonly used in biology, at least, to basically create some semi-sterile conditions in the lab. Basically, what it does generates a flame on the top, and this flame here is used to sterilize different things, but just by putting it inside the flame. For example, you can put your flask, your Erlenmeyer flask, you can sterilize the neck of it just by putting it on top of it for a few seconds. This way you make sure that basically kill all the bacteria that are present here outside before you pour the media inside the blood. This can be used to sterilize many other things like pipettes and so on. Now, the other advantage is that around this Bunsen burner, basically you create a semi-sterile area, because basically the area around here becomes hot. By becoming hot, it tends to go upward. You're basically creating a draft of hot air going upward. So meaning that if you're on top of it, anything that falls that's light enough on top of it towards the flame, towards where you were working, will actually be deflected and brought up again. So this area here is basically semi-sterile, meaning that if you work close to the flame, you have a much less chance of contaminating whatever you're working with. Okay? Now, let's go back. So now you have your media, you cool it down to 55 degrees, you have your Bunsen burner, you have your plates, which have been labeled. Now you're ready to pour the media inside the plates. The way you do it is you'll take your 500 milliliter Erlenmeyer flask, you open up the top, you put it, quickly sterilize here by going through the flame, and then you take one plate at a time, you slightly open the lid, as you can see here, with your left hand or right hand, one of the two hands, and you pour enough of the media from the flask in a way that you cover the other half of the plate. Okay? Once you've done that, you close the lid, you put this plate aside, you take another plate, you do the same thing. Slightly open it up, pour some media, first always put the flask on the flame, and then pour. Now you try to work as close as you can to the flame. So in this way there is a very little chance of contaminating the plate when you open it up. After this, then you'll have your 5 plates, and you will let them stand there. Do not shake them at all. You let them stand there for 10-15 minutes on your bench with the lid partially open, and then you'll see that your media will solidify in there. So at the end of this process, you'll have your media ready, your sterile media ready to be used to plate the bacteria. Now, how do you plate the bacteria on that? You'll be provided with a culture of Escherichia coli, so bacteria that normally grows in your intestine, so you have to be very careful with them when you work with them. So what you have to do is, using a pipette, you take 0.1 milliliter of the bacteria that you've been provided with, and the other hand you slightly open the plate, as you can see here, and you pour this 100 microliter into the plate. You see that basically this 100 microliter was that only on some part of your plate, will not spread evenly. Okay, so now, since you want to make sure that you grow as much bacteria as possible, you want to spread this bacteria in a way that they will cover the whole plate here. So how do you do that? You do that using a spreader or a hockey stick. This is a piece of glass which has been bented, so in a way that now you have a surface here which can be used to spread anything with. With this spreader, before you actually use it, you have to sterilize it. You want to avoid contamination of your plates with bacteria which you do not want. You want to grow only the Escherichia coli, nothing else. So to sterilize it, first you put the hockey stick or spreader inside some ethanol or isopropanol, and then you put it on top of the Bunsen burner. This will basically flame all the ethanol here and basically will allow to sterilize this part of the hockey stick or spreader. Now you let it cool down for about 20-30 seconds, keeping the spreader is always close to the flame, so there's a less of a chance to get it contaminated again. And then you can finally spread the bacteria which you have added in this step by moving it, basically spreading it. I'll show you in the lab how to do it properly. Basically by spreading it up and down, and slightly rotating with the other hand the plate. Once you're done that, you have spread your bacteria. You do the same thing for the other 4 plates left there. Once you're done with that, finally we'll collect all the bacteria, which would then be put in an incubator at 37 degrees, like the temperature you have in your guts. Obviously, Escherichia coli likes that temperature, and the plates will be inverted. So basically the lid will be put on the bottom and the media on the top. This will favor the bacterial growth. So the media will be on the top, this will be in the media, your bacteria are here, right below, and we'll put like that on the incubator to let them grow. And then the following week, we will use these bacteria, we'll learn how to harvest them, so basically to collect them after they have grown, and we'll proceed from there. There's a series of safety things that we'll discuss in class before we start, so you can read them right now at the end of the screencast, but we'll discuss them in class, so we'll make sure everyone is safe during this experiment. Alright, so we are all done, and I don't know anymore how to stop this, but we'll do it. See you in class.